Hello, in this podcast we're going to be looking at DNA replication, and we will understand how DNA is replicated. First, a couple of review things. The shape of DNA is a double helix. This is different from what we're going to be seeing in the diagrams because it's just easier to see it as a two-dimensional straight ladder. Just keep in mind it isn't. Its actual shape is a double helix. There are also 4 bases, and those are the parts of nucleotide that change. And in those 4 bases, there are 2 couples, where adenine loves thymine and cytosine loves guanine. You need to keep those in mind in terms of the replication. First, I want to show you an experiment that was done by Meselson and Stahl. This was very important in terms of finding out how DNA replicates, because there are a few models on how DNA can replicate. How do we end up with 2 strands of DNA that are identical to what we had before? One model that we see here is this called semi-conservative replication. In that case, what happens is the new strand has one old strand and one new strand. The old strand is from the original double helix, and the new strand was constructed to make the new one. Then there's conservative replication, where the old strand is used as a template to make the new strand. By template, I mean it's used as a basis for copying. But when the new double helix is made, it's made completely of new material, and the old DNA strand is maintained. And then there's the last model is dispersive replication, and for that, each DNA strand has a mixture of old DNA and new DNA. Now I'm going to show you a video that shows you the Meselson-Stahl experiment.
The Meselson and Stahl experiment provides evidence for semi-conservative replication of the DNA molecule, where the 2 parent strands serve as the template for synthesis of new strands. In this experiment, bacterial cells were grown for several generations on a medium containing a heavy isotope of nitrogen, N15. The DNA in these cells therefore contained heavy N15 nitrogen. The cells were then transferred to a new medium containing the normal lighter isotope, N14. At various times after the transfer, samples of the bacteria were collected. The DNA was then extracted and dissolved in a solution of cesium chloride. The samples were then spun rapidly in a centrifuge. When the cesium chloride is centrifuged at high speed, a concentration gradient is established in the tube. DNA molecules move in the gradient until they reach a place where their density equals that of the cesium. DNA containing N14 moved to a position in the gradient determined by its density. DNA containing N15 is denser than that containing N14, so it sank to a lower position in the cesium gradient. After 1 generation in N14 medium, the bacteria yielded a single band of DNA with a density between that of N14 DNA and N15 DNA, indicating that only 1 strand of each duplex contained N15. After 2 generations in N14 medium, 2 bands were obtained, one of intermediate density, in which one of the strands contained N15, and one of low density, in which neither strand contained N15. Meselson and Stahl concluded that replication of the DNA duplex involves building new molecules by separating parent strands and then adding new nucleotides to form the complementary strand on each of these templates.
So to summarize, here are the results of the experiment. In a 0 generation, all the strands of DNA are made of the heavy nitrogen. So it comes out as 100% in heavy. Second generation, we see that all of them are medium, and that tells us that when you have a medium, that means that 1 strand is of old DNA and 1 strand is new DNA. And you see how you go from 100% medium to 50%, each time it's half, 50%, 25%, 12%. And then the rest of it goes into the light, where the strand is all new DNA. So that tells us that throughout this whole experiment, generation 0 through 4, there are 2 old strands of DNA, and there remain 2 old strands of DNA throughout it. So this type of replication is semi-conservative, where the old strand is maintained and it serves as the template or the model for copying the new strand of DNA, which then attaches to the old one. Now I'm going to show you how the DNA replicates. First step is that an enzyme will break the hydrogen bonds between the bases. If you remember, the hydrogen bonds are right here in between the bases, and remember, they are very weak. The bonds between all the atoms in each of these molecules is strong, so those won't break. So an enzyme comes along and unzips it, and it unzips it really from one end to the other, where it's going to come out sort of like this. Can't show this to you here. All I can do is just simply move it apart. And then another enzyme comes along and it brings in the copies. So you see A doesn't come in through here, so that's no good. I'm just going to put this aside. Down here, this would be a C. C goes with G. So G comes in. Give it a moment. So it comes in. Then the G here goes with the C. And also on the other end, G here goes with C. C goes with G. G goes with C. I'm just going to continue doing this end here because it's so much quicker. T goes with A, and now I'm going to use this A. A goes with T. A goes with T again. Oops. G goes with C. And T goes with A. And the same thing occurs along here too. I'm just going to do maybe one more because it's going to take quite a bit of time to put this all together. And as the stuff is being put together, sometimes a mistake will occur. And there's another enzyme which will come in usually and correct the mistake. Sometimes that won't happen, and if that happens, then you have a change in the DNA sequence, and that's known as a mutation. So we end up with two strands of DNA. Here's one, and this one here will be built. With this one here, I'm just going to do the rest in a little snap. And through the magic of editing, we have two complete strands. And as you can, and over here we have the old strand, and here's the old strand. And now we've got the two new strands attached, and we now have two exact replicas. And as you see here, if there was a mistake done in the copying, that'll be known as a mutation. Now we come to the concluding questions for the vodcast. And for these questions here, you need to refer to the diagram at the left, which is taken from a Regents exam. So for question 1, what is the sequence of strand X? And for this, I want the sequence of the bases. You just need to put down the letter. Number 2, what happens at point 2 during replication? Number 3, does the DNA replication that we have in this vodcast, does that occur during mitosis, during meiosis, or both? And number 4, how does DNA replication maintain the same DNA sequence? So that concludes our vodcast, and I'll see you in class tomorrow.